Have you ever wondered how cancer develops at a genetic level? This is a question that scientists have been trying to answer for decades, and while we haven't cracked the code entirely, we've certainly made some headway. Today, we're going to delve into the intricate world of cancer genetics. It's a complex process, but understanding it is key to developing more effective treatments and ultimately saving lives. In the coming minutes, we'll explore the foundations of this process: the cell cycle, genetic mutations, oncogenes, and tumor suppressor genes. Each of these elements plays a crucial role in the development of a cancer cell. Our journey will take us from the normal functioning of cells to the point where things start to go awry, leading to uncontrolled cell growth. Strap in as we unravel the genetic intricacies that give rise to cancer. To comprehend cancer's genetic basis, we need first to understand the cell cycle. It's a series of phases that a healthy cell undergoes: the resting phase, or G0, the first growth phase, G1, the synthesis phase, S, the second growth phase, G2, and finally, mitosis, or M. At each stage, checkpoints ensure the cell's integrity, preventing errors that could lead to problems down the line. Now, what propels the cell through this cycle? The answer lies in proteins, specifically cyclins and CDKs. These act as the cell cycle's drivers. For example, when cyclin D binds to CDK4 or CDK6, it triggers a series of events that lead to the S phase. However, not everything always goes to plan. Disruptions in cyclin-CDK levels can upset the balance, causing cells to grow in an uncontrolled way. And this, my friends, is a hallmark of cancer. But what instigates these disruptions? Enter genetic mutations. These are changes in the DNA sequence that can alter the function of genes. They can occur in various forms, each with the potential to transform a normal cell into a cancerous one. Point mutations, for instance, involve changes in a single nucleotide, the building block of DNA. This seemingly small alteration can have profound effects, changing the amino acid sequence of a protein and thus altering its function. DNA amplification, on the other hand, leads to an increased number of copies of a certain gene. This can result in the overproduction of proteins that drive cell growth and division. Chromosomal rearrangements involve parts of chromosomes breaking off and reattaching elsewhere. This can lead to the creation of a novel gene with a new function. Lastly, epigenetic modifications don't change the DNA sequence but alter gene expression, switching genes on or off. One such consequence of these mutations is the activation of oncogenes. Oncogenes, such as the RAS and MIC genes, are pivotal players in this transformation process. These genes, under normal circumstances, control cell growth and division. However, when they mutate, they turn into oncogenes, instigating a cascade of uncontrolled cell proliferation. Let's take a closer look. Oncogenes are like a car's gas pedal stuck in the go position, driving cellular growth and division in an unregulated manner. This is due to mutations that lead to their perpetual activation, effectively bypassing the cell cycle's checkpoints. Prototypical examples are the RAS and MIC genes. When mutated, RAS proteins remain perpetually active, continually signaling for cell growth and division. Similarly, a mutation in the MIC gene leads to its overexpression, driving excessive cell proliferation. In this relentless drive for growth, oncogenes play a pivotal role in the transformation from a normal cell to a cancerous one. On the flip side, we have tumor suppressor genes, which act as our cell's guardians. Tumor suppressor genes like p53 play a crucial role in preventing cancer. These genes are the cell's guardians, standing as a bulwark against uncontrolled cell growth. They respond to DNA damage by initiating pathways that halt the cell cycle, giving the cell time to repair its DNA, or, in extreme cases, inducing apoptosis, cell death, to prevent the propagation of harmful mutations. However, when tumor suppressor genes are inactivated, which can occur through mutations or epigenetic modifications, these safeguards are removed. The cell cycle is no longer regulated as it should be, leading to uncontrolled cell growth. In essence, the cell's brakes fail and it begins to divide uncontrollably, forming a tumor. This is the dark side of our genetic machinery, Mutations that activate oncogenes or deactivate tumor suppressor genes can tip the delicate balance of cell growth and death, leading to cancer. The interplay of these genetic alterations, activation of oncogenes, and inactivation of tumor suppressor genes gives rise to cancer. So, let's tie it all together. Today, we've journeyed through the complex world of cancer genetics, unraveling the fundamental mechanisms that underpin this devastating disease. We began with the cell cycle, a series of phases that every normal cell undergoes. Here, we saw the importance of checkpoints that maintain the integrity of each phase, ensuring the orderly progression of cell growth. But what happens when this process goes awry? Proteins, specifically cyclins and CDKs, are the conductors of this cellular symphony, driving the cell through its cycle. However, when these proteins lose their balance, it can lead to uncontrolled cell growth, a key characteristic of cancer. We then turned our attention to the role of genetic mutations, those subtle yet significant changes in our DNA that can transform a normal cell into a cancerous one. These mutations can activate oncogenes, like the RAS and MIC genes, leading to a state of perpetual activation that fuels uncontrolled proliferation by bypassing the cell cycle's checkpoints. On the flip side, we have tumor suppressor genes like p53, the guardians of our genome. These genes respond to DNA damage by initiating pathways that halt the cell cycle, facilitate DNA repair, or induce cell death. But when these genes are inactivated, the safeguards are removed, and abnormal cells are free to proliferate unchecked. In essence, cancer arises from a delicate interplay of genetic alterations, activation of oncogenes, and inactivation of tumor suppressor genes that allow cells to evade regulatory mechanisms and drive uncontrolled growth. Understanding these mechanisms is not merely an academic exercise. It's a crucial step towards identifying potential targets for therapeutic intervention, providing hope for more effective treatments in the future. Thank you for watching. Your journey into the genetic underpinnings of cancer doesn't have to end here. If you found this video informative, don't forget to subscribe for more content. Until next time, stay informed and empowered.